Targeted covalent inhibitors are a class of small molecule compounds that can covalently bind to conserved amino acids of specific target proteins. The bond functions as a precision lock, leading to the sustained inactivation of the targeted protein. Targeted covalent inhibitors are composed of an affinity component and a covalent warhead. The affinity component initially binds through non-covalent interactions, forming a preliminary complex. The warhead subsequently establishes a covalent bond with a nucleophilic residue in the target protein, enhancing the binding affinity and ensuring sustained inhibition. Typical discovery strategies for targeted covalent inhibitors include the ligand-first, fragment-based, and electrophile-first approaches. The design of general covalent drugs involves 4 key processes: target identification and residue selection, hit identification, binding characterization, and lead optimization. Covalent warheads are the essential reactive groups in a drug molecule that form covalent bonds with the target protein's amino acid residues. In covalent inhibitor development, warheads are categorized by the residues they bind to, with cysteine targeting warheads being the most common. Cysteine residues are relatively rare, making them highly selective targets for covalent interactions. Common warheads feature structures such as amides or sulfonyl fluorides with conjugated unsaturated bonds, strained small cycles, and functional groups with unsaturated carbon-nitrogen or carbon-oxygen bonds, such as cyano and aldehyde groups. Common acylation techniques are widely used to covalently modify parent molecules containing amino groups. Some examples require the aldehyde fragment to form an alpha, beta, unsaturated bond, via Novo-Nagel condensation. The sulfonyl fluoride is another classic covalent warhead. Easy-to-handle products such as sulfur fluoride exchange and AISF allow chemists to pick their preferred method to modify the parent molecule on the oxygen-hydrogen or amino group. Additionally, the halogen exchange reaction of sulfonyl chloride provides another robust pathway to access aromatic sulfonyl fluorides. The unsaturated carbon-Q bonds can be typically accessed via the dehydration, hydrolysis, or reduction, oxidation of corresponding precursors. For boronic acid warheads, synthesis typically involves introducing boronic esters, followed by hydrolysis. As these are common functional groups, all the classical synthetic strategies can be adopted and work well on their fitted scope of substrates. For the introduction of strained small cycles, it can take several steps of chemistry conversion to synthesize a common intermediate that contains the reactive warhead and to use the intermediate to derivatize the parent molecule. Since the strained small rings are reactive, more caution needs to be paid to the handling of these unstable intermediates. WuXi AppTec Research Chemistry Services has built a comprehensive covalent chemistry discovery platform offering synthesis, analysis, and purification services for covalent inhibitors. Our team of over 1,000 experienced synthetic chemists has successfully delivered projects for over 100 clients. Since 2021, we have synthesized over 40 distinct types of warheads and delivered over 180,000 covalent compounds. For further questions and requests, please feel free to contact us.